In ScarfWeb, you can do differential gene expression analysis comparing any cell you see in the UMAP. In this video tutorial, I will show you how to use the lasso tool to compare clusters, visually determined groups of cells, cells based on gene or cell surface protein expression, or cells of different source. Here we are on SCARF's analysis interface with a site-seq stem cell dataset that is actually 4 leukemia patient samples that have been merged. So please check out our dedicated video tutorial for how to merge datasets. You find the lasso tool up to the left of the UMAP. When selecting this feature, you can now select any cell or group of cells within your UMAP or T/S plot for further analysis. We start by dividing up the red cluster 1 into 2. We start by selecting this bottom part and save it under Save on top of the UMAP. Next, we select the top part of the same cluster. Now, to remove contaminating cells from other clusters, we can click Adjust and remove these here. Afterwards, we save, and now we have 2 populations that we will soon compare. Next, I will show you how to compare 2 different clusters. You can use the lasso tool to select just a few cells of the cluster you're interested in. Next, you click Adjust and include all the cells of this cluster. You save, and then you do the same for any other cluster that you want to compare this with. Now let's see how you can select and compare cells based on the expression of a gene or a cell surface protein. We have a site-seq dataset, so I will select based on cell surface protein expression of c-KIT. I first highlight c-KIT in the UMAP. I then select the cells that I want to include in the analysis. And then I go to Adjust and remove cells from contaminating clusters. Then I click Adjust again and select Feature. Now here I got an histogram with c-KIT expression. And by dragging the handles, I can select, for example, the highest c-KIT expressing cells within my selection. And you can in parallel see in the UMAP which cells are selected. When you're satisfied, you will confirm and save your selection. Now I do the same thing, but select the lowest c-KIT expressing cells. As I mentioned in the beginning of this tutorial, this dataset is actually a merge between 4 leukemia patient datasets. I will now show how you can do gene expression comparison between different samples within the same cluster. First, we go to the metadata feature and select Source Dataset. On the left, you will now see the different sources, and if you hover the mouse over these, the cells from each sample will be highlighted in the UMAP. Now we use the lasso tool to select cluster 1. We can clean up the selection under Adjust. Confirm the selection and go back to the Adjust feature. Under Cell Metadata, you can now select only the cells from one sample. In this case, I select patient 3. Confirm and save, and do the same for the sample you want to compare with. Under Saved Selections, you can now see all the selections we made during this tutorial. If you click on the eye, you will see them highlight in the UMAP. If you want to do differential gene expression analysis between these, you then click Compare and select the 2 cell selections that you want to analyze against each other. You can do several comparisons at the same time without losing any speed. It will take ScarfWeb a couple of minutes to finalize the analysis. When the analysis has finished, you will get a few alternatives on how to visualize the data. You can either choose the table feature, which will present a table with the most upregulated genes for each cell selection. You can copy the gene names or download this entire table for downstream analysis. If you expand this table, you will also get more statistics for these genes. For perhaps a more visually attractive output, you can choose to see this data in volcano plots. When you click the volcano plot button, you get a pop-up window with the data from a comparison. You can highlight your genes of interest, and the plot can be completely customized using the feature on top to adjust fonts, size, and labels. When you're happy with your plot, just download it. The downloaded data from ScarfWeb can be directly imported to your presentation or your manuscript figures. With ScarfWeb, you go from data generation to data figures in just a few hours. I hope this tutorial was helpful. Don't hesitate to reach out to me directly on goran.karlsson@nigen.io or visit our webpage nigen.io. if you have any questions or feedback. Good luck with your single-cell genomic analysis.